talking about the sequence-dependent properties of DNA. But before we go ahead, we need to all get at the same base. So we'll start with the struggles of understanding DNA, and then we'll understand their properties, and then we'll go to computational modeling, and so on and so forth. Don't worry, the slide will keep appearing again and again to let you know you've completed the modules and you feel happy about it. Okay, so let's start. So the history and chemistry of DNA. If you ask me which would be a word to describe DNA, I would call it rebellious and resilient. From the 1700s till 1950s, people believed that protein was the genetic material. In 1953, Hershey and Chase did an experiment where they had bacteriophages which had radioactive phosphorus and radioactive sulfur. Radioactive sulfur in case of the protein core, and radioactive phosphorus for the DNA. They put them in live mice cells, centrifuged them, and then wanted to check, do they find radioactivity or not? Where they had radioactive sulfur, they did not find any radioactivity in the cells, but in case of radioactive phosphorus, they found radioactivity in the DNA in the cells. That indicated that the genetic information transfer happens from one human to another, from a virus to human, or any other means by DNA, implying DNA won the ground. DNA is the genetic material. Okay. Now, in the same very year, 1952, 1953, people in Cambridge, Watson and Crick, and at Imperial Rosalind Franklin were trying to figure out the structure of DNA. Okay, we knew DNA existed. Is it a helix? Is it a globular form? What is exactly it? So they did some extra diffraction experiments, and they found that DNA is a B-form helix, which has a right-handedness. It is made of 4 bases: adenine, guanine, thymine, and cytosine, with a backbone. The phosphate group has 2 strands running from 3 to the 5' end, where one has a free phosphate, other has a free OH group. So, well, the good story was we figured out DNA is the genetic material. We found the chemistry of DNA, and we found that it mainly exists in the double-stranded form. Now, you might hear that the best friends are those which stick around during bad times. Let me tell you, double-stranded DNA is not a best friend of its own counterparts. Okay? So whenever there is recombination, there is replication, there is transcription, they get divorced. Right? They become single-stranded, and the proteins go there and they start working. Now, it is very important. These are exactly the situations which can change or make the process of creating new human beings, because during transcription, your proteins do the work. During recombination, genes are transferring from one to the another. And during this duration, you find small DNA constructs in your body. Now, another important off-topic, these pictures have been generated using VMD. You all are VMD experts, and I'm sure you can make better pictures than this. Now people got excited. Okay, DNA does exist in the body. We know a bit about double-stranded DNA. But you have to understand, so far, people had been studying proteins. They were developing techniques to understand that proteins were the genetic material, and crystallography techniques were so good that people could understand and study proteins easily. But now, DNA came into line. So it stole all the light, and now people had to start working on experimental techniques to understand DNA. That meant people had to start from scratch all over again. Some of the pioneering works was done by the group of Carlos Bustamante, as Chris pointed, where they used optical tweezers to understand the force versus extension response of double-stranded and single-stranded DNA. Now, the best way to understand is, imagine a piece of thread tied to one end of the floor. At the other end is an optical tweezer, and you start pulling it. You slowly start pulling it from a force going somewhere between nothing to 70 piconewtons or 80 piconewtons, and you measure the end-to-end extension. When you do that, now on the red curve there, you see that's double-stranded DNA. Double-stranded DNA has a hysteresis curve. Single-stranded DNA is a limitless polymer. You keep extending it, keep extending it. It doesn't break apart. So technically, single-stranded DNA has a persistence length way lower than of a double-stranded DNA. What do I mean when I say persistence length? In the words of Tamara Schleck, whose DNA mechanics book, if you've read, she says persistence length is the length of a polymer up to which it appears straight. That means If you were to look it from top, you wouldn't be able to tell if this polymer bends or not. So for double-stranded DNA, that length is 150 base pairs or about 50 nanometers. For single-stranded DNA, that's about 1.5 nanometers. That means it can form loops, it can form secondary structure, but for double-stranded DNA, that structure is well defined. Then some work was done by small angle scattering experiments by Hal Liffert and people who tried to understand the mechanical response of homopolymers. What do I mean homopolymers? Homopolymers are poly A, poly T polymers. So you're just using A and only T to form these long polymers. Good. We understand a little bit about DNA. Not as much as proteins, but we're trying. Now, so you've understood a bit about DNA. So we can now start getting into where does computing come into DNA? I like to call this the teppanyaki science or the salad bar science. Imagine you go to a teppanyaki restaurant where you select your bowl. Your bowl is the size of the salad that you're going to get or the size of your meal that you're going to make. Then you go to the next counter, you select your proteins. You go to the next counter, you select your sauces. And then you go to the cook and you say, mix everything up, right? Your meal is ready. Something similar is building up coarse grain or DNA modeling. You select the resolution of your model. You decide if I want a 1 bead per base pair model, I want a 2 bead per base pair model, or do I want to go higher resolution depending on the problem that I want to solve. Then you decide what is the problem that I'm looking to solve. Do I want to look at the thermodynamic properties of DNA? Do I want to replicate the elastic mechanical properties of DNA? Or I just want to understand it as condensates, just as a charged polymer. You define, therefore you decide the properties that you want to do, which is your protein. The next is choosing the source. You then define what are your potentials going to be. Will you use a Finney potential? Will you use a harmonic potential? Will you use a Morse potential to define how your DNA is going to behave? And then finally, you cook everything up and you either get oxDNA, you get mRDNA, depending on the choices you make. oxDNA is a top-down model where 1 bead per base pair is the only and the minimum and maximum resolution that you can get. It was parameterized again for Santa Lucia nearest neighbor modeling. Nearest neighbor experiments where basically they try to understand how stacking of A over T or G over C changes the thermodynamics of DNA. Then is the mRDNA model, which I think Chris has spoken in great detail. Something that you all will resonate with is all atom structure. I think yesterday Diego was presenting one of those pictures. And when you talk about all atom DNA, there are many force fields. There is BSC1, there is CHARM, there is DE Shaw, there's OL15. And all these force fields, most of them have been parameterized against quantum mechanical calculations. People perform simulations on quantum calculations on very small DNA strands and try to develop the parameters so that the properties could be replicated. And some of the people who have been doing pioneering work on these are Modesto and Cheltenham. Okay. Now, if I were you, I would ask the question, well, you have so much on the market. Which one do I pick to solve my problem, right? The answer is, you can question it two ways. One is, can any problem be solved using any of the methods? No. Can a problem be solved in any of the ways? Depends on the problem. The answer may be yes or no. For example, a collaborator reaches out to you. He says, I have a DNA origami, and I have to submit a review or a response to the editor within 2 weeks. I want you to get the configurational analysis, right? What would you do? I would either do oxDNA or mRDNA. I would prefer mRDNA because I can do that in a day and get over it. I would do oxDNA in case the collaborator says, I'm not sure if my origami is stable. I don't know if it breaks apart. So I want to measure the thermodynamics and see how many base pairs are breaking. Now, if your collaborator says, I want to submit the paper in Nature, you put the system on all atom simulations, put 100 dependencies on Frontera and go on a trip to Hawaii. Enjoy. Come back and you will get a big simulation which has been sorted for you, done for you. And you can do your post-processing and analysis thereafter. Now, the important point is, can I solve any problem? No. Can I solve some part of a problem using one method and can I go to the other? Yes, but let's take it this way. You all are at the workshop today. Some of you took a flight, some of you took the bus, some of you drove, some of you mixed matched the means of transport. But you could bridge from one transport to the other, or you knew the method of bridging. Now, when you want to solve a problem using multiple methods, the problem can often be bridging. You did some simulation in all atom, now you feel, oh, okay, I got something and now I want to go back to coarse graining because I cannot solve this problem using all atom. You need to back the coordinates and the potentials back to reproduce those problems. You did something coarse graining and you want to go in detail. So for that, with Chris, we have developed mRDNA, which you can use going from oxDNA to mRDNA to all atom, or all atom to oxDNA. That therefore means that bridging becomes easier. So an example would be, say your collaborator or you decide you want to understand the properties how engineered proteins change DNA origami. How would you do that? You would study an engineered protein with a small section of the DNA or a DNA origami. You would get all the potentials, get all the properties, parameterize, and come back to mRDNA. How would that help? Since you have got the interaction at the lower level, you can use IBI, come back, and then do your dynamics of the origami and understand how, in general, that small interaction that you were able to understand in all atom can be fed back and can be used to understand it way quickly than putting 100 proteins on this origami and making a 20 million big system. Now, in case you were wondering why this wasn't moving, this simulation has been submitted. If you get back to me in 2 months, I will send you a movie. Okay. So that's about bridging models. And so, okay, we've understood DNA. We've understood why computing comes into play. Now, let's try to understand what are the experimental advances that people are making in understanding single-stranded DNA. Now, there's a small story about this project. This project started about a year and a half ago. I was a freshly minted PhD student, and a German came to us, who is a faculty at TU Delft, with this plot. That's it. And he said, hey, Alex, we have 1,024 single-stranded DNA sequences. Can you simulate them in all atoms? I was on the Zoom call, and I vehemently shook my head and laughed. Little did I know, next day I was sitting in Alex's room and said, you are going to do this project. So lesson, never say never. Okay. So the technique is SPARCS, which stands for Single Molecule Parallel Analysis for Rapid Exploration of Sequence Space. What really happens? Now, when people do sequencing of DNA, they can only do it for a few, or if they do FRET analysis, they can only do it for a few. So Chomlin developed a technology. What it does is, you have billions of sequences. You have a library of billions of sequences, and you put them on a flow cell. So you select some sequences, you immobilize them, you put them on a plate. Next, you go to FRET. You put that plate onto FRET. You get the FRET measure, and then also you get the coordinates of each of the sequences. You put the same plate, you sequence it, you get the coordinates. So you have three parameters. You have coordinates, you have FRET measurements, and you have the sequence. You can backmap all of them onto one plane. That means for all of the sequences, you have the measure of FRET, you know what the sequence is, and you know where they were placed. That means you can do that for any salt, any sequence, and generate all data. Now, yes, we're talking about FRET, we're talking about measurements. What is FRET? FRET is fluorescence resonance electron transfer. It is a non-radiative transfer where there is an acceptor and a donor. So the donor is an electron donating group, acceptor is an electron accepting group. The distance between the acceptor and donor should not exceed beyond 10 nanometers. If it is beyond 10 nanometers, you will not get any intensity at FRET. That also means FRET is directly proportional to the distance between the acceptor and donor by the factor of r to the power 6. That means if your acceptor and donor are placed close, you will get a very high intensity of FRET. As soon as you place them far apart, your intensity of FRET reduces. That implies you can study the secondary structure which is based on distance property. And this works beautifully for DNA because DNA is a homopolymer made of just negatively charged nucleotides. Nucleotides are all negatively charged and they hate each other. They don't want to be together because it's all negative charge, which doesn't want to be there. So now we understood what FRET is. Now we knew what the experiment was, and we wanted to simulate it. So what did we do? So this was the experiment. They have the sequence. They had one end of the DNA bound to the slide glass. They had an acceptor, they had a donor, and they had a sequence, 5 nucleotide sequence of DNA in the center. We made the structure in OxDNA and we simulated it, right? So you put forces at one end of it, you simulate them. Now in OxDNA, you can only do this for poly A and poly T sequences. Why? Because OxDNA was parameterized for poly A and poly T using FRET data. You can try using it for poly G and poly C, but do not come back telling you don't get answers, because you won't. And we were able to convert the donor acceptor distance into FRET using this formula, where R is the Foster radius related to the FRET efficiency, where the FRET efficiency is roughly 50%. And you get a distribution like this. So this is where the experimental FRET was. This is the OxDNA FRET. And this is very similar looking to the experimental data that we have. Now let's understand the properties of poly A and poly T. If you look at the sequence, if you look at this chemical structure, adenine is a double ring. It has a hexane and a pentane. Here is only a hexane. If you understand chemistry, you will understand that rings which are bigger in size or which are more rings attached to single one, they like to stack more because there's more pi stacking. They can basically get energetically lower and stabilize themselves by stacking. As compared to poly T, which is a single guy roaming around, he finds it difficult to stack, and therefore it behaves like a freely jointed chain going haywire. That means you would observe a varied distance in stacking in poly T as compared to poly A, where you see a very sharp curve. That means double rings like to stack, single rings don't like to stack, right? So, and the good part is we are able to do this all using coarse grain simulations in a matter of 1 day. Now, okay, we understood the importance of poly T, poly A sequence dependence. Now, I will also talk about sequence average. This part will repeat again when I tell you about why I did this. So we tried to understand if you do not discriminate in the DNA in terms of sequence, what does that mean? This model of DNA was developed mainly to understand nanotechnology. It was initially developed to understand DNA mechanics, and then it developed to understand nanotechnological devices, where you have big random sequences and you do not really matter, it doesn't matter much what your sequence is. So you want to just understand the dynamics, and it kind of fades away when you have such large structures. Therefore, you do sequence averaging. What does that mean? If you take up a nucleotide, it will not exhibit properties of A, T, G, or C, but it will exhibit combined properties of A, T, G, C. So technically, you average over your parameters. And therefore, we observed that when you plot the acceptor-donor distance versus the 1,7 nucleotide. So remember, there was an acceptor-donor distance, and there was a small single-stranded DNA sequence in the center. So you plot the correlation between the distance of 1,7 nucleotide and acceptor-donor distance, you realize poly T is spread out, doesn't like to stack. Poly A is center, likes to stack. And when you see sequence average, it shows properties which are kind of between poly A and poly T. Importantly, with poly A and sequence average sequences, you observe that they also show hydrogen bonding. Why? Because you have A and T in the system, and they would like to bond together. And the right-hand side of the plot is when you take small, so you have data here going from 0 to 8 nanometer. Now you end up taking small bins, each of 0.1 nanometer, and you average over y-axis. That is to basically show you the trend in the variation of distance for acceptor donor and 1,7 nucleotide. This will be used again, but I will tell you why we did this. Now, We all have been going to parties, right? It has been fun. Let's go to a DNA party. What is a DNA party? A DNA, A, T, G, and C. I'll be slow because I wanted to go back to the initial frame. I couldn't find any good emoji for it, so I put my own face. A, T, G, and C. If you go to a party where people don't like each other, what happens? You try to stay away, right? Nobody wants to talk. Suddenly, some extrovert walks in who is also friends with A and G. They start talking. They start coming close together. More sodium walk in. They start coming more close together. Suddenly, people who could not fit in a room are suddenly all together in a small space. This is exactly how salt determines the properties of DNA. As and when you start increasing salt, DNA becomes compact. DNA becomes rigid. And that means if you were to measure the distances at high salt, you would see lower distances. If you go at low salt, the DNA is very spread out. Now, can you see?
Yeah.
Sorry. Sorry? So, okay. So we've been to the DNA party. Now let's get back to work again. Now, the question is still hot. 1024 sequences. How are you going to characterize what's really happening at the 1024 sequence port? Therefore, we use computational microscopy, as Klaus would say, or all atom MD simulations to understand what are the interactions that actually drive the FRET values that we get. So, what did we end up doing? We had to do all atom MD, but the problem was the existence, non-existence of all atom force. Now, as I told you, why did I start my talk with telling you the development of DNA? DNA is a very recent molecule. It has failed to gather as many experiments as proteins have. Therefore, that means that when people were developing force fields for DNA, force fields are not bad. You don't have data. It is a loop. It is a machine learning problem. You don't have good data, you don't have good models. Therefore, most of the data that you have is modeled against human calculations. Now, while people are doing experiments, you constantly see assessment, evaluation, progress, modification of force fields for DNA. Now, this is not to scare you from doing nucleic acid chemistry, okay? This is to tell you you have to make an informed choice when you choose your force field. It becomes very important because when you choose a force field which overestimates your certain properties, you will get garbage. If it underestimates, you will still get garbage. Therefore, you constantly need to apply CU fixes, MD fixes, or use experimental data to use gold bars or restrictions to get your desired interactions that you want to study. And that is exactly what we did. So what we did was, firstly, if you did end up putting the whole system that experiments, or we did it in OxDNA, this project would take a lifetime. And as Imad said, time scaling is a problem in MD. If you can solve it, you can get a Nobel Prize. I think it's really something you should grab. You should try doing that. So we couldn't do that basis, but we can shorten it. And that is why we wanted to get the correlation between the acceptor donor distances and 1,7 nucleotide. If we can target the distance that we got for the smaller construct, we can use it as a code bar. We can put a distance restraint, and the FRET distribution that we got can be used to calculate the force constant of that restraint. What happens? When you put a distance and a force restraint and a force constant with a restraint, you limit the interactions to an experimentally observed quantity. What does that mean? If you put this structure in all atom and let it run, you will obtain nothing. Done that. But if you put the distance and if you put the force constant, which you can measure using this Kohlbar equation, which you can measure using this force constant equation. Be careful, in Kohlbars there is a half in kx square. When you use NAMD, it doesn't have a half in harmonic potential. So you have to be careful about it. So when you do that, you can extract all data that you need. Now, single-stranded DNA does exist in nature, does exist in solution. What stabilizes it? What are the interactions? The very first interaction, as it is DNA, is stacking interaction. Stacking is pi-pi interaction where the two rings are over each other, right? Now, we were able to observe from all atom simulations that purines stack better than pyrimidines, which was a straightforward chemistry answer. But we observed that heterogeneous purines stack more than homogeneous purines. What do I mean by that? Poly A will stack less than AG. AG is equal. Any guesses why? Electrostatic repulsion. If you put the same molecule over each other, it has the same electron cloud. Despite the fact it wants to stack, it will remain stacked. If there is a cloud which has a similar kind of ring, but not exact electron cloud, it can remain stacked for longer because electrostatic repulsions would fall away later. Similarly, does it just stack against its nearest neighbor? No. Single-stranded DNA has a persistence length of 1.5 nanometers. That's about 3-4 bases. That means it can bend. That also means that you can see stacking beyond the nearest neighbor. And therefore, wherever there is more stacking, there is less next nearest neighbor stacking. These are two of the major interactions that basically would stabilize single-stranded DNA. Next, hydrogen bonding within the chain. Single-stranded DNA can bend. Single-stranded DNA can form these crazy kind of structures where the first nucleotide goes between the fourth and the third one and can form hydrogen bonds. And you can do these by measuring the acceptor-donor distance and angle between your bases. So, cool. We understand about single-stranded DNA. We got a little ambitious. We're like, okay, we can do these systems. Why not go to something complicated? We wanted to understand the dynamics of Holliday junction. Now, what is a Holliday junction? Holliday junction is a 4-way junction of DNA that occurs naturally in your body during replication. And it was actually coined by Robin Holliday, who was a British biologist, if I'm correct. But it did not gain much, it did not gain people's attention. Ned Seaman came up in 1982 with the concept of DNA origamis. And he said, we cannot make big origamis if we take a single strand or a double strand of DNA. But if we branch out DNA, we can make very big origamis. And that is when Holliday junction made their way into the synthetic world. But the dynamics is very interesting. Holliday junction has 2 isomers. Now, to understand the isomers, have a look at the blue strand and have a look at the yellow strand. Blue strand stacks over the yellow strand. In this case, the red strand stacks over the green strand, and it goes via the open state. At low salt, as I told you, DNA doesn't like itself. It will open up. It will try not to stay together. As soon as you have high salt, it likes to stack. Now, the interesting stuff is, people have proved that the sequence at the core decides which isomer is going to be more stable, or which is the isomer that is going to be predominant. That is dependent on AG stacking or AT stacking or GG stacking. Again, the same thing which applies to single-stranded DNA now becomes more predominant because you're talking about 4 bases stacking. Now, if you put this structure in all atom simulations and you increase the temperature, you might see this. But it will give you nonsense after a while because DNA will break apart. Okay? If you go beyond 380, 390 Kelvin, you will start seeing thermodynamically impossible structures in this. So we tried to use a Kohlbar, which was based on contacts between the 2 strands. And if you go from one to the other, you can get a free energy plot, which basically tells you that for this specific sequence, this isomer is more favorable than this isomer, and it goes via the open state. You can do this for as many sequences as you want. It roughly takes 7 days per sequence because you have to do multiple replicas and you have to do them for very long so that you can fill up your energy space. Takeaways. Takeaways are we are making strides while trying to understand single-stranded DNA with techniques like SPARCS, which can also be used to understand properties of holiday junctions. Secondly, as Chris mentioned, if you do coarse graining diligently, if you choose your potentials properly, if you choose your degrees of freedom correctly, you can really decipher a lot about DNA in a span of a day or two. Thirdly, all atom MD itself for single-stranded or double-stranded DNA can help us invade certain problems. But if you want to do problems for which data is sparse or for which single-stranded force fields are not developed, you have to use experimental data. You have to combine atomistic simulations with experimental data to completely understand the problem. And most importantly, metadynamics can also be used to understand the sequence-dependent properties of holiday junctions. I would like to finish my talk by thanking everyone in the XMT lab, especially Chris and Alec, and our funding agencies, NSF, NIH, and STC. I hope they continue to fund us like this. And supercomputing resources, and obviously the Jew lab, who have provided us with wonderful data. And these are certain links that you can use, and we have to take questions.
So you use these constraints in the coarse grain type model, but you can also use those constraints and limit unnecessary excursions in the regular all atom system also.
Coarse grain you can't do it. Because coarse grain, the potentials that you develop for G and C, they will not give you the correct measure. Because you are actually using that to get the distance from a sequence average.
So, I mean, we know in chemistry that some of these aromatic ring systems, they stack in two forms. One is planar stacking, one is perpendicular stacking. Do you observe any of those?
So this one specifically is planar stacking because I put a condition of 30-degree angle. So if it goes anything beyond 30-degree, I will screen it out. And that can be counted as, not the perfect stacking. But actually, we are not going to follow these norms of stacking. We will be measuring this as dry stack. So we will measure the number of water molecules that you find between the two stacked molecules to actually measure whether they are really stacked. The stacking is a dry stack.
The stacking that you showed differences that are sequence dependent, like for A versus T, is that usually always stacking of the bases in a facial fashion, or can there be like edge to face stacks that take place?
The same question.
Yeah.
In this case, we did observe it, but I don't know if I have that as a hidden slide. I measured it as perfect stack and not so perfect stack, right? Because when you talk of, okay, you are talking about that thing. When you talk about stacks, mainly when you see stacking, people talk about protein ligand, that is the major concept of 5 angstrom distance and 45-degree angle. Single-stranded DNA, you have to understand it's connected by a backbone. It cannot do whatever it wants, right? If it does like this, it will break apart. So you have to be careful. So you do not observe completely like this. Yes, but you observe certain kind of deformation, and that is why we're going to rule out this. We'll do complete dry stacking measurements.
So what level of theory?
Sorry, what level of?
Theory. And how do you decide on that? Is the types of interactions present influence each other?
So all atom simulations or all atom MD simulations are basically solving Newtonian mechanics, the Verlet scheme for all of these atoms, right? So how do you decide? Well, the parameters that exist, for example, I know this is recorded, so I have to be careful what I speak. If you talk of force field, there are certain force fields which overestimate the stacking. So certain potentials are developed. For example, what happened was people started doing simulations, they started getting data. As and when they started getting data, they started manipulating their force field. So new force fields came into question. Now, certain force field will reproduce certain properties very well. So it's a matter of choosing. For example, if you want to study a structure which you know never breaks and is very stable, go for DE Shaw or Amber. If you know something is crazy, don't go for that. Because probably your structures will never see fraying or breaking in certain force fields, but those are real phenomena. Because single-stranded DNA is just an electrostatic bomb. Doesn't want to be together.
So, when you're using ARBD, what is the going from the simulation time to the real time? What is the time that you can?
So we are talking of time scales of picoseconds for DNA and similar scales for ARBD, because if you go to less, you will never be able to simulate. If you go to large, you'll explode the system. So we're talking of picoseconds, 10 raised to the minus 12 for ARBD or coarse graining, and 10 raised to the minus 15 for all.
I was just wondering about, the previous approach was using this, iterative Boltzmann immersion. Like, do you use such approaches?
I haven't used for this, I've used for another project, so that we can talk about it. Where we had these DNA cubes and we simulated them in a force field called X. And they did not behave. Then we did them in a force field called Y, we behaved well. But we had a lot of those structures and we couldn't afford to do all of them in one. So we took this, we did IBI and developed potential. So now we can pick up any structure of that size and use the potential that we developed for that. So basically, yes, we have done that. For this one, it did not make sense because we could reach a system size which could be resolved.
Last thing, I have one last message before we go for the coffee break.